Hey guys, it's Ryan, and in this video we're going to look at the taste pathway. So anything that has to do with taste, of course we have to start with some food or some source of taste. So let's say we just ate a meal and we have a bunch of food particles floating around in our mouth. Now, those food particles alone won't give us any taste sensation. If we have no saliva in our mouth, we're actually not going to be able to taste anything because these food particles are too large to be sensed. So what has to happen are these food particles have to be digested, and then the little chemicals have to dissolve into the saliva and then travel to the taste buds. And these little particles, these little chemicals, are called tastants, not to be confused with the larger food particles that they're coming off from. So before we go any further, let's just get oriented with this diagram. This is the side of a lingual papilla, which would kind of wrap around here. And I always thought that the lingual papilla was the taste bud. The little bumps on our tongue are the taste buds, but that's not true at all. They're actually lodged in the epithelium, usually within the wall, the side wall, of the bumps on our tongue, on the papilla. So this is the papilla, and this little bulb structure is the taste bud. Alright, so if we go back to the tastants, they're going to dissolve through the saliva that coats this lingual papilla, and they'll do so at different rates, depending on their molecular weight and so forth. This is called temporal dispersion. So there's a time element to how they disperse through the saliva. And they'll eventually reach a structure just referred to as the taste pore. So that's just the sort of hole, if you will, in the epithelium that these tastants can penetrate to reach the taste bud. These little extensions, these little hairs, are actually microvilli of the taste receptor cells that we'll talk about in a little bit. So these are sometimes referred to as, I think, taste hairs, but we'll just call them microvilli, and they're part of the taste receptor cells. And that's kind of our that's probably the biggest deal of this whole pathway, or the taste receptor cells. And that's because they're the initial peripheral sensory cells, the sensory apparatus that's going to detect the tastants and tell us something about the food that we're eating. So these taste receptor cells, there's a lot of stuff going on. One thing to note is that because they're located so close to this outer wall, to the epithelium, and they're of course vulnerable to any sort of insult. So they usually have a turnover rate of about 10 to 14 days. And so you have these little sort of stem cell-ish cells called basal cells that can step in and differentiate into new taste receptor cells if they're damaged or if they get exfoliated or something like that. So let's say that we had some tastant and just a general pathway that we're talking about now. It would be accepted by some receptor on these microvilli that would then initiate some response within the taste receptor cell. We're going to say it's a depolarization, and that would cause this taste receptor cell to release neurotransmitters. And it could be any one of, there's usually 40 to 60, I have like 5 drawn here, but any one of the taste receptor cells of the entire taste bud could release some neurotransmitter. It could be acetylcholine, it could be glutamate, it could be serotonin. Those are a couple of them that you'll see here. And it would eventually interact with a nearby first order neuron, the first afferent neuron in this pathway, and it would initiate an action potential if there are enough neurotransmitters, if there's enough of a response. It'll initiate an action potential, which we know will travel throughout this neuron and continue on the pathway. So let's define. So we said this was a first order neuron because it's the first actual neuron in this pathway. Of course, we had the taste receptor cell, but we can't call that a neuron. So we'll start here, and this is usually, we're generalizing a little bit, but this is usually part of the chorda tympani nerve, which we know about. We know it's a major nerve involved with the taste sensation and also salivary secretion. But here we're talking about a whole tract of this nerve is dedicated to a whole bunch of taste receptor cells leading to these first order neurons all throughout the tongue. And they're all kind of traveling through this chorda tympani. And you'll notice that the cell body is located halfway through the neuron. So there's an axon here in this half, which we call the peripheral axon. You can think of that like it's going towards the peripheral sensory cell. It's going towards the outside. And then this lower half, in this case, of the neuron is called the central axon because it's headed towards the central nervous system. I think that's a good way to look at it. And this kind of neuron is called a pseudo-unipolar neuron. Maybe not important for this class, but just a good thing to note for the structure. That's why there are kind of these two axon branches coming off. And the cell body is hanging out in a ganglion, which is a group of cell bodies in the peripheral nervous system, and it's the geniculate ganglion, which is located in the skull, lodged in the skull, geniculate ganglion. And you can think of this as sort of similar to the dorsal root ganglion for touch sensation. It's like the dorsal root ganglion, but for taste. Okay, so once we go through this first order neuron, then we're going to get to a point where we're no longer in the peripheral nervous system, but we move to the central nervous system. And we do so by entering the brainstem. And specifically, we're going to synapse in the solitary tract nucleus, or nucleus of the solitary tract. And this is located specifically in the medulla of the brainstem. So now we're at the second order neuron. This is always the case, that the second order neuron is the one that crosses over. So it's going to cross the midline as it ascends, and so we'll say this is maybe the midline of the body. If it's starting off on the left side, it's going to end up on the right side of the brain in the thalamus. So this is where the second order neuron will synapse with the third order. And the third order will send branches to two different locations of the primary gustatory cortex, which makes sense. That's the principal place in the brain for taste to be integrated and modified and understood. And this will happen in the insula of the brain, which you'll remember is lodged in the folds of the temporal lobe of the brain, sort of in that lateral groove area, and that's for the actual detection of the stimulus. And the other branches will go to the parietal cortex, specifically the center. You can think of the somatosensory, the primary somatosensory portion of the brain. Actually, let me just pull up a picture here so we can look at it. If you'll remember this sort of diagram, if we're looking at where the tongue is sort of on this lateral portion of the parietal lobe, and remember the primary sensory processing area is located on the posterior side of the central sulcus. So it's going to be part of the parietal lobe, and specifically on the lateral portion of it. So that's where all these fibers are going for the parietal lobe part. And for the parietal cortex, that's going to be sort of not so much detection of the stimulus, but more, do I like this taste or do I not like this taste? And of course, there are more complications to that. There are more processing and modulation that goes on that we don't really have to worry about too much. All right, let's just mention while we're here at this tongue diagram that there are a couple of different nerves that are involved with this this pathway, and specifically, there we go. We have the 7th cranial nerve, the 9th cranial nerve, and the 10th cranial nerve. And this is for the anterior portion, anterior 2/3. Then we have the sulcus terminalis, the posterior third, and then actually behind the tongue, at the root, the base of the tongue, we have the vagus nerve. So these are 3 different cranial nerves that are involved in carrying taste sensation. And then I just want to briefly mention that each of these taste receptor cells will have different affiliations for different classes of taste. So some of them might have a lot of receptors on them for salty tastings, and maybe a couple for sour, or any mix in between. So there'll be a good way to think of it is they'll have different sensitivities to different tastings. And one taste bud could have all different taste receptor cells. It doesn't have to have just one type. So let me just briefly go over the different taste classes. So we have salty, and if we have this temporal dispersion of tastings, the first thing that'll happen once they reach receptors on the taste receptor cells is that, so you can think of salt as typically NaCl, and the Na plus will simply just go through sodium channels on those receptors. And you know that once sodium goes through and enters a cell, there'll be a depolarization event, which we typically associate with activation. And once we have that, we'll have an influx of calcium into the cell, and this will eventually result in neurotransmitter release, and then this action potential that goes through the pathway. Sour is very similar to salty, except instead of going through sodium channels, instead of the NaCl, we have an acid. So some citric acid or something. So we're going to have a lot of H plus with acids. And this H plus can block potassium channels, which will halt any hyperpolarization. So if we don't have hyperpolarization, we'll naturally get depolarization. And so we get the same sort of response, the same ultimate response from that. And now for, I'll just move over here, for bitter, for sweet, and for umami, which is like a savory steak or something like that. These 3, which you can think, I just remember bitter, sweet, and then you can add in umami. Is kind of a helpful way to remember them. This pathway is a little bit different, where we don't have an ion channel, we have a G protein coupled receptor. And so the tasting will bind to the G protein coupled receptor, which will activate phospholipase C, which will cause an increase in inositol phosphate, and this will lead again to calcium influx and then neurotransmitter release. So I hope you found this video helpful, and I'll see you all next time. Thanks for watching.